Whoa there, Harry. You sure are consuming a bunch of phenylalanine.
Phenol, what? All I'm doing is drinking some Diet Coca-Cola.
Phenylalanine. It's one of 20 amino acids that we use to synthesize protein molecules. Of these 20 amino acids, phenylalanine is one of the 7 that are essential. This means we must obtain all our phenylalanine from foods such as nuts, animal products, and artificial sweeteners, because our bodies cannot synthesize it.
Wait a minute. So if I need phenylalanine to make protein molecules, shouldn't I get as much of it as I can?
Not quite. Because phenylalanine is an essential amino acid, it must be strictly controlled. If too much phenylalanine is present, the amino acid can become toxic and causes neurological damage. On the other hand, a lack of phenylalanine leads to decreased synthesis of proteins and neurotransmitters. The amount of phenylalanine is controlled by an enzyme called phenylalanine hydroxylase.
So let me get this straight. We need phenylalanine, but it has to be just the right amount, and so we use something to control that called phenylalanine hydroxylase?
You got it. Phenylalanine hydroxylase is an enzyme whose responsibility is converting phenylalanine to tyrosine by adding a hydroxyl group to the aromatic ring. The PAH gene, which is responsible for making phenylalanine hydroxylase, can be found on the long Q arm of the chromosome 12 between positions 22 and 24.2.
Okay. What does this phenylalanine hydroxylase look like?
We can actually look at the structure of phenylalanine hydroxylase in 4 different ways. This picture shows the primary and secondary structures of the enzyme. The black letters in the picture show the series of amino acids that make up the enzyme's primary structure. Above those letters is the secondary structures that are produced as a result of the bonding between neighboring amino acids. Each molecule of phenylalanine hydroxylase consists of multiple beta-plated sheets, which are indicated by arrows, and alpha helices, which are shown by spirals. Next, we have the tertiary structure, which is made by folding the secondary structure. Within each molecule of phenylalanine hydroxylase, there are 3 domains: an N-terminal regulatory domain, a C-terminal tetramerization domain, and a catalytic domain. The N-terminal regulatory domain, residues 1 through 142, is shown in yellow and contains an autoregulatory or N-terminus sequence, residues 19 through 33. Meanwhile, the C-terminal tetramerization domain, residues 411 through 452, is shown in red and is responsible for interlocking 3 additional molecules of phenylalanine hydroxylase into a tetramer. The catalytic domain, residues 143 through 410, is the purple portion of the enzyme and the center of catalytic activity. It has an iron ion, shown in green, at its center, which is important for enzymatic action. This domain acts as a hydrophobic cage for the phenylalanine substrate. The last structure is known as the quaternary structure, which is made by combining multiple tertiary structures into a larger unit. In humans, phenylalanine functions as a tetrameric unit. This means that 4 molecules of phenylalanine hydroxylase, which are held together by their tetramerization domains, combine and function together. These pictures both represent tetrameric forms of phenylalanine hydroxylase. However, the one on the left shows the cartoon structure, which highlights the alpha helices and beta-plated sheets. Meanwhile, the picture on the right is a space-filling model.
Whoa, okay. So it looks like phenylalanine hydroxylase is a very specific structure. But how does it change phenylalanine into tyrosine?
The hydroxylation of phenylalanine to tyrosine requires both tetrahydrobiopterin, or BH4, and oxygen, and occurs during 2 main steps. First is the oxidation of the BH4 cofactor, which creates a reactive hydroxylating intermediate, and second is the entrance of oxygen into the amino acid substrate. Once phenylalanine hydroxylase has been activated, BH4 binds rapidly, which results in the phenylalanine hydroxylase 117 BH4 binary complex. Once phenylalanine enters the active site, it binds to form a ternary complex to which O2 rapidly binds.
Created using Powtoon.